Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week, I'm with Marcin Paduch, Head of Platform at Metaphore Biotechnologies. Marcin talked to us about his childhood spent surrounded by scientists and doctors and how that led him into a career across structural biology, protein engineering, and groundbreaking biotech startups. We also talked about the importance of staying curious, embracing technology, and being open to unexpected opportunities. I'm back on Careers in Discovery with Marcin Paduch of Metaphore Biotechnologies. Marcin, welcome to the show.
It's a pleasure to be here. Thank you.
Fantastic to see you. And I am fascinated to start by talking a little bit about Metaphor. I'm really intrigued by the concept of biomimicry. Some of our audience, I'm sure, will be familiar with it. Some may not be so much. But if we could start out by talking a little bit about what you're up to at Metaphor and the work you're doing there, that'd be fantastic.
Of course, yes. So at Metaphor, I serve as a head of platform. So I'm overseeing efforts of the entire team to build the MIMIC platform. This is our acronym for it.
Yes.
And the goal is to really design new and improved therapeutics that will maximize the patient impact. And how are we doing this? We're leveraging machine learning. Then we are using the concept of mimicry. And mimicry as such is probably, as you know, one of the oldest tricks in nature. And It's effectively a way of gaining adaptive advantage. And we're using this concept to really try to solve a very tough therapeutic problems. And then how does it work, right? We are focusing on pharmacophores of interaction. So we are taking a very deep dive, looking very carefully on how given molecule is interacting with its natural partner.
Okay.
And by doing so, we are mapping residue by residue, atom by atom, the interaction itself, isolating key features, the key hallmarks of this interaction, and also what makes this interaction between receptor and ligand, for example, functional, what makes that molecule really tick. And that pharmacophore effectively can be stamped on any therapeutic modality you would like to pursue, given the product profile you want to accomplish. And the process itself, since this mapping is so comprehensive, not only allows you to retain the hallmarks of interaction and its functional properties that not naturally occur, this is what we mimic, but also improve on that. So we can build a drug which can have improved potency, or the drug which has improved half-life, or the drug which actually has some of the side effects of natural molecules that occur in nature eliminated due to ability to really fine-tune at the level of detail necessary to do so.
I see.
And this machine learning enabled approach is enabled effectively by recent advances in AI/ML field, in combination with diverse tooling of protein engineering tools. And then here we use tricks known to every protein engineer. So a lot of combinatorial screening, a lot of NGS, a lot of work on cells, and really working on molecules in their most natural environment. So that's the metaphor in a nutshell.
Yeah, fantastic. And so you're essentially looking for things that already have the interactions that you want to leverage,
Exactly.
and then using that to your advantage to
Yeah.
deliver different deliver different molecules, deliver different payloads, deliver that kind of thing.
Because naturally, molecules really evolve to facilitate those interactions. They are essentially building blocks. So if you look at this Lego block, it will have those little fiducials, these dimples. They're meant to interact with another Lego block. So we're exploiting this idea of taking the original Lego block and converting that Lego block into the new entity, new molecule, where we can fully realize potential of that molecule. And that's the power of the concept.
I see. That's a great way to think about it, actually. And presumably the principle of
Mhm.
the technology is applicable across a huge range of targets and disease areas and all this kind of thing. Are there particular areas that you guys are focusing on to start with?
Obviously, as you know, we have announced recently the collaboration with the Novo Nordisk. So we
Hmm.
have a standing interest in cardiometabolic space, obesity as well. That's one. We have also, with the entire team, have a continued effort to build out our internal pipeline.
Yes.
And this is where we're focusing on several different classes of molecules. One of them is inflammation immunology. And we see, the growing interest and growing need really to exploit the idea of mimicry or the mimic
Mm.
platform for engineering molecules that can really deeply impact the signaling pathways. The molecules that can be surgically precise at targeting or triggering certain receptors,
Right.
but also, and there's a new concept, multi-targeted. So can we actually take multiple molecules and can find one singular solution that
Mm.
actually does multiple functions? And obviously, this concept is not new in small molecule world because people work with, let's say, kinase inhibitors. It's
Yes.
fairly rarely exploited in biologics. Obviously, multi-specifics are there, and then complex constructs are there. What differentiates us is that we look really at engineering singular molecule which has single binding interface that accomplishes diverse functions.
Yes.
And that's where Mimic platform comes in.
Yeah, I see. And you mentioned a couple of things when you were describing it that I just wanted to pick up on. So the first thing is it's a very old trick in nature, as you said,
Yes.
and we see it in predators and all these kinds of things, right, across the every ecosystem.
Yeah.
But you mentioned as well that then it's the impact of machine learning and AI that's kind of allowing you to leverage that. I'm assuming that's in terms of being able to accurately map the molecules that you want to mimic and then recreate them. Is
Right.
that
Yeah. So, normally when you work in protein engineering, right, most people are familiar with approaches like phage display, ease
hmm
display, ribosome display, etc. And those approaches were, developed and organically grew from maybe, colony picking analyzers to NGS, right? And the fact that right now we can so deeply sequence the resulting repertoires makes those approaches very amenable to machine learning
right?
because it's
Yes.
humanly impossible really to comprehend the data set of that size. And the power of the approach is that we really try to combine the very thorough experimental approach where we generate very robust data packages that
hmm
inform the function of the molecule, inform the sequence and design space, and use the ML tools to really find what makes the molecule tick, what are the patterns you observe, and what are relationships you can infer from data sets you're seeing that really tell you about this is structure, this is sequence, this is function, how
Yes.
those things are combined and merged into one. And that's, how we go from, the Google fold of possibilities to something that you can manage and handle. And that's the beauty of this pharmacophoric mapping.
No, that makes sense. And I think it's fascinating the way that across, lots of areas of drug discovery and development that the technology we have now is really enabling us not just to come up with new things, although that's one thing, but also to leverage things that are very, very old and that we know work to different ends.
No, this has been a pattern in industry of doing this because
Mm,
they're well designed technologies, well tested concepts that
yes.
just need a new approach, a new angle, right? And image analysis as such, really sparked the revolution in machine learning and AI. And that's being broadly applied to many scientific concepts, right? If you astrophysicist, you know a lot about image analysis. If you were old school physicist working with structural biology, let's say you work with cryo-EM technologies,
Yes.
now you have all those amazing tools to come up with averaging and come up with the molecule maps that otherwise were not possible. And this is why the field is booming, right? And now the next frontier is probably how to combine those new approaches in AI ML with structure prediction and experimental validation into one.
hmm
And I feel like, at Metaphor, we have the technology that allows us to do this.
No, that's great. And it's a fascinating concept, and I have lots more questions, but in the interest of our listeners being here to hear about you, Marcin, I'm going to park those for now. We may come back to them. So I'm always interested as well in the origins of where people's careers come from. And I'm assuming you
Yes,
didn't just wake up one day and think, do you know what? I'm going to use biology and machine learning to mimic
yes.
cells. I'm sure there's a
Oh
journey to
no.
this point. Um, why, we're going to go right back, but why science in the first place for you,
Oh,
and why drug development? Why this career? Do you think? Where does it come from?
I think it's a great question. I feel like I had no choice. I was really destined to.
Okay.
And my story is that I grew up in a family of scientists and doctors. My dad
Mm,
was a doctor, MD, PhD. My mom was math and physics teacher. And what was fascinating at the time was growing up in a health resort in Poland,
mm,
and my dad was running a hospital attached with this health resort facility where people would come for treatments after injuries, after complex
ah,
surgery,
yeah,
or with chronic disease. And he had a lab, like diagnostic lab, associated with his function. And there was no after-school programs at the time. So would I go and hang out? Well, I would stay with my dad. If he was
yeah,
available, I was free, I would stay in his office, do my homework. If not, he will kick me out, and I'll go to diagnostic lab to bug all the technicians.
yeah.
And you are 10 and you learn, okay, this is how you measure hematocrit. This is how centrifuge works. This is
Hmm.
how you look at the erythrocytes and count them on the microscope. So this was like my introduction to science, or science that translates from scientific concepts to medical interventions, right? And that
Yes.
was the piece. And then experience with my mom was also interesting because, again, if I was not hanging out with my mom, I will go and hang out with my mom. And she had, since she was teaching physics and math, she had this demonstration room on the back
Mm,
where all the instrumentation was stored. So there was everything there, from electrostatics to magnetism to a full-size diesel engine if you want one,
fantastic.
and like a chemicals, analytical instruments, everything. And there were only two rules: don't make any noise and don't break anything. So, with the childhood like this, right? I wanted to be a doctor for a very long time. I didn't get accepted to medical school and
Mm,
biotechnology program, but ultimately, what got me fascinated about staying with, applied science and then really biotechnology was the fact that I was really interested in how things work.
right.
What is the mechanism, how things function, and this fascination in structure function, in how molecules work, how to design drugs, pushed me really to start very early working in the lab. And I
Mm,
started really as a computational biologist. And this is,
okay.
connection what I do at Metaphor.
Yes.
This was my humble beginnings, learning how to simulate molecules, learning how
Hmm.
to work with structures, and that morphed very quickly into me realizing that computation was not ready to answer a lot of questions at the time.
Yeah.
And I moved to biophysics, structural biology. I moved to protein engineering. And I, as effective, am a trained structural biologist. I work in structural biology for 4 years in drug design. But what really was, maybe formative or really informed my career was this step into protein engineering.
Yes, I see.
And this, built in me this fascination about the design space, about, treating the molecules, the biological molecules, as collection of the building blocks
Hmm.
that you can have all the fun possible, replacing components and figuring out what they do.
Yeah, I see. I see. So yeah, as you say, there was a hint of destiny about the whole thing from a
Yes.
very early
There was
age.
an element of that, yes.
Yeah.
And I went from effectively structural biology then to hardcore protein engineering. I did my
Hmm.
PhD in molecular biology and biochemistry, and I found and joined the laboratory of Tony Kosyakov. And Tony was a long time director of protein engineering in Genentech.
Hmm.
He was there from early days,
Yes.
and from the times where the company was still hosted in the warehouse in South San Francisco. And what Tony taught me was how to take advantage of a scientific freedom and
Mm,
freedom to do anything with the time you have. And the second one, how to leverage collaborations, how to work with people.
Okay.
And what was maybe characteristic of my, very short postdoc stint was that there were essentially revolving doors on collaborators.
Mm,
Tony would bring, two, three people a month to work with, every person coming with a different scientific problem to which we were really applying protein engineering and structural biology at the time, to really crack it, to
okay.
get deeply, right? And this exposed me to like a different style of work where to make it work, you need to dive in deep, understand the problem, and enable the collaborator.
Yes.
And and for me, this was really an introduction to how you work in industry. Because
Yeah.
the greatest work in industry is done on high-powered teams, the teams that everyone contributes, everyone brings their best, and really makes things work in a, well, limited budget, accelerated timeline with a lot of good innovation, right? And
Yes.
this training, lasted for me for about 10 years. I moved on, maybe from a postdoc-like role to a more
Mm,
manager role, and started working on consortia that were really blending the, academic work with industry-sponsored research.
yes.
And this is where I got exposed to working with big pharma companies. This is where I learned some of like business development chops, how to land the deal, how to sell the project to the partner,
Mm,
how to get through the due diligence. This was a very invaluable experience. And the other aspect, which I don't want to maybe short sell, is getting all the operational experience. Because
right. Okay.
I went from essentially scientist and a postdoc to someone who has to run the efficient lab. So I
Hmm.
need to learn about budgets, how to report on progress, how to build and negotiate equipment, how to get contracts in place, how to hire people, how to keep them happy, right? And that training was effectively a start of my scientific career in industry.
Yes. So you packed quite a lot into that 10-year period.
That was a
And
lot, right?
and even in the postdoc, what a fascinating way to learn the principles, because you're learning all of these principles, but also you're being
Yeah.
asked to apply them to lots of different practical problems
Yes,
that
and
others are facing.
exactly, and it builds in you the hunger for different fields,
Mm,
for really understanding the diverse set of problems you may be working on.
yeah, yeah, absolutely. So it sounds like you were perhaps prepared more than most for taking that step into industry at the point that you did, having sort of been involved in partnerships, collaborations,
Yeah.
consortia, as you say, and even to the point of business development. Um, Tell us a bit about those first steps then to Grail initially.
Yeah, so Grail came almost out of nowhere. I got
Mm,
recruited to join the team, and really the ask was, can you come in and build the lab of the future?
right.
And effectively, I got wedged between very strong team that came from Illumina, strong NGS scientists, people who know everything about sequencing and genomics, and extremely strong software team that was sourced from many startups or many tech companies in Silicon Valley.
Hmm.
So if you think about this, it was almost 50-50 split between like a
Okay.
Grail and Illumina. And my
Yeah.
role was effectively figure out the language you need to speak when you work with software engineering and data science, and figure out the language you need for the genomics and blend them into one strategy so we can make progress on developing innovative product.
Yes.
And as a part of the product development team, we were effectively building the foundation of what is now the Galleri product, which
Hmm.
is one of the few multi-cancer detection tests out there. It was a fascinating experience because, at the time there were no tests that were detecting cancer at fidelity and also diversity of cancers that can be detected.
Mm,
And my exposure at the time, which I found very valuable, is essentially getting acquainted with regulated environment.
right.
What are the requirements of building something that was going to be informing patient treatment? How to document the work you did, how to validate an assay, how to convert the assay into efficient, automated form of that assay that can be run at low cost. Because in
Mm.
the end, right, to reach broad population of patients, you want to build an assay which is affordable, which can
Yes.
be covered by insurance, and that assay has to be not only affordable but has to perform. And that was very, very crucial learning from me because I went, from early phase drug discovery to
Mm-hmm.
diagnostics. And that, that I considered it a sabbatical was for me the moment where, I started to really appreciate the regulated environment,
Yes.
really appreciating why it's done. And then, connection to this diagnostic lab from my childhood was important because I grew up, seeing how that is used, right?
Yeah,
How that
absolutely.
influences the medical decisions people are making. So the transition for me was fairly natural.
Yeah.
And another piece that I really enjoyed is was working on inception and then sample processing for
Mm-hmm.
large scale population level clinical trials.
Yes.
So that gets me exposed to electronic data capture. That gets me exposed to, how do you essentially build software systems that are very well meshed with reporting, with data collection, with analytics. So that was a fascinating experience. Nonetheless,
Mm.
it was a sabbatical, how I see it,
Yeah.
because right after that, I went straight into discovery, and I took the risk and joined a small neuroorganoid startup in San Francisco. And
Mm.
what got me enticed at the time, I walked in an interview and landed on the parking lot, and I didn't know where the company was. It was essentially was
Okay.
like a door in a garage. You walk into the garage, and there is a full-scale clean room in there with robotics
Wow.
and people working hard on the problems, right? So this was, an experience, right? So this was really garage state startup, which was building a high tech stuff,
Yes.
and it was fascinating. It was essentially a relatively a short experience, but very valuable because I got to
Mm-hmm.
experience to work with small molecules and also help to formulate pipeline and then get acquainted with, styles of work and then types of applications you have in neuroscience.
Yes.
And neuroscience is a very complex and amorphous beast, as you may know.
Absolutely. Yeah. And then building organoids for neuroscience is adding to that complexity.
Of course, right? And the
Yeah.
model of neuroscience are so needed. So there is a clear
Yes.
need, and effectively a very under resourced area of science, right? So
Yeah.
I'm very much looking forward to, what that's going to happen in neuroscience field. That definitely needs more innovation. Um,
Yeah, and I think it's interesting to see there's now more funding, right? Especially over the
yeah.
last couple of years, there's more progress starting to be made, more progress certainly in terms of potentially being able to penetrate the blood-brain
Uh-huh.
barrier and solve some of those
Yeah.
big problems. And I think,
Well,
yeah, these are things that are, and these are conditions that are probably going to affect, similar to cancer, they're probably going to affect the majority of us in some way, right? So it's a big problem to solve.
yeah, and I'm sure that we will be making progress in
Mm,
neuroscience, because I think neuroscience as such, will definitely then benefits from depth and breadth of data you're acquiring,
mm,
and then it needs to be very strong in imaging and genomics and really learning about structure of brain and structure of neuronal connectivity that otherwise, it's very hard to tackle, right?
yes.
Observing things at the resolution necessary to build a drug in neuroscience and then having a translation from, almost a petri dish model to an organism,
Yeah.
that's a great challenge.
Yeah, absolutely. And it's interesting the journey you've been on during that time, I guess, because you've arrived at Chicago as a scientist, as a postdoc, as you said, as you talked about, you've learned to run an efficient lab, and the differences between those two things. Um, what a great pitch Grail gave you to come and work for them, by the way. Come and help us build the lab of the future. I might have to steal that one. Um, which is, that's going to appeal to someone like you, right? Who wants to see how
Yeah.
things work and get into things. And then, but also going into diagnostics, as well as the connection it had to
Yes,
your early experiences. I guess the value model is different in diagnostics, right?
it
You
is.
have to be efficient because you can't charge $1 billion for a drug
Of
and
course.
all this kind of stuff. Um, and then this kind of almost speakeasy
Hmm.
automated lab that you've you've then gone to is another fascinating departure. But
Yeah.
then, sort of, as you say, you've gone back into the therapeutic field at this point, the drug
Hmm.
discovery field at this point. So take us through a little bit the next couple of steps
Yeah.
that lead up to where we are today.
Yeah, so I think after that I went to Arsenal Bio, which
Mm,
is a premier cell therapy startup. I joined Arsenal as a director of technology development, and for me this was effectively going back to my protein engineering roots,
yes.
with a strong systems biology influence.
Hmm.
Um, so really taking advantage of my know-how and knowledge how to build effective lab, how to automate workflows,
Yes.
and how to protein engineer components that you normally would want to engineer your cells with, right?
Hmm.
Because CAR Ts as such, effectively a heavily modified product, and that you are building a hybrid or chimeric or fully synthetic molecule that's decorating those CAR-Ts.
Mm,
And then the synthetic biology component comes from how do you really wire signaling pathways to make effective therapeutic. And the challenge here is obviously on how the modification happens, how you edit, and then also how do you manufacture the therapeutic at scale, because the
yes.
cell therapy manufacturing is a large problem, because everything becomes custom product delivered to a patient
Right.
that needs to meet very high standards and is very costly to produce.
Hmm.
So a lot of companies actually working on how to improve the engineering process, as well how to improve the manufacturing process.
Mm,
And
hmm.
and I'm rooting for cell therapy. I think it's a great field, and I think we will be seeing a lot of innovations coming from cell therapy field.
Yeah.
And the what led me to Arsenal was really this combination of science and technology.
Hmm.
So how to apply the concepts I learned at Grail at System One, concepts I really developed for protein engineering and my experience in working consortia, and apply it to developing cell therapy product.
Yes.
And and this was fascinating. After that, I went to Xylus, which is again organoid startup. Uh,
Hmm.
and this time cancer organoids.
Yes.
And
Okay.
the startup was founded by three people, two faculty from Duke, Shilling Shen, and as well Hans Klevers,
Hmm.
the godfather of all organoids. And this was, very interesting technology platform for effectively matching patients to therapy and
Mm,
building the microorganosphere technology platform that allows you to really screen the drugs at scale, or screen
yeah.
the drugs at throughput and fidelity, allows you to make relevant decisions. So that was very interesting. Nonetheless, I felt like I departed again from my molecule design, protein engineering area, and effectively got recruited to join Metaphor.
Yes.
And Metaphor, I feel like it's this binding glue of different experiences, right? Because I no longer work on setting up and solving hard problems in protein engineering, computational biology, but also can have a broader impact, effectively building a business.
Mm,
So how do
hmm.
you recruit? How do you formulate your pipeline? How do you manage the teams? How do you motivate people? How do you get things underway, and things that are, effectively very hard, and the things that were not done before? And that's,
Yes.
the ultimate startup experience that many of us seek.
No, absolutely, absolutely. And I think looking at the journey you've taken through your career, obviously you've not looked for the easy problems to solve, which I think is important, right? They've all
Yep.
been pretty new, novel ground, breaking into new
Yeah.
areas. Um, and looking at your progress through your career as well, you've progressed really steadily, really rapidly, and
Yes.
in each role, in terms of responsibility, in terms of the breadth of focus. Um, what are the things do you think that have enabled that to happen, enabled that progress to happen in your career? What do you think has led to that
Um,
for you?
I think one thing is the ambition, and then
Hmm.
staying focused. That's, probably a common component. And the second piece that I cherish is um, time to think and think deeply about what you do and why. Also, allocating the time in your schedule for self-reflection.
Mm.
Really taking a step back from this day routine and really think about the interactions you have with people, the way you handle tasks you are being asked to perform, and really getting into really building out your soft skills,
Right.
building your ability to sometimes lead by influence, and that's what was, I think, most effective, right? I took part in a lot of different training opportunities where
Yes.
you can hone the skills like this. I always allocate some time to listen to podcasts,
Mm-hmm.
listen to books when I'm driving.
Yes.
So I think in terms of my career progression, this is one of the enablers. So not only science skills and exposure to technology, but also learning how to work with others,
Yeah,
because
absolutely.
that's the piece that frequently is omitted. Because when you are growing as a scientist, right, it's natural that you progress to your scientific career by the sheer power of your intellect,
Right.
and that's different than the what actually makes industry work well, which is very much focused on collaboration.
Yeah.
And developing those skills is a different set of skills that frequently, when you go through your science training, you're very much unprepared for.
It
So
is.
that's essentially a self-training. Also essentially time that you need to allocate in your schedule to actually get better at.
Yeah. Yeah, completely. And I think, people are moving into, we're moving into an industry where everybody is really, really smart. So are you going to necessarily be the smartest person? Well, maybe, but it's a pretty high bar, right? So you've got to look for different ways to differentiate yourself as well. And as you say, it's often a lot of the surrounding skills that make a big difference. And I think before I move on to this next bit, actually, you
Mhm.
mentioned podcasts and books and things like that. Any particular recommendations for people building their career in biotech right now?
There are many. It's more of like what you're looking for,
Yeah, yeah.
right? And there are a lot of books that I recommend to different people depending on the problem you want to solve. I'm a big fan of listening to books, and I'm
Mm,
subscribed to all the libraries I've been
yeah.
close by to. So nothing in particular, right? But
Okay.
it all depends where you are.
Keep learning though, and keep finding new perspectives and new
Yeah,
opinions. Yeah.
it's
Um,
more exposure, right? Exposure to sometimes different way of thinking.
Yeah, and I think a lot of people are sort of on this self-development path, whether, as you say, through podcasts, books, courses, whatever it might be. But the other thing you talked about there, which I thought was really pertinent, was this idea of reflection and taking time to think and reflect and um, really sort of consider where where you are, where you're going, what the important things are. And the reason I pick up on that is that I think it's we've become as a society so sort of short in our attention spans that actually being able to take a step back and reflect without having to look at your phone every 2 minutes and all this kind of stuff, it is becoming a competitive advantage, I think.
Yeah, and that's the time that many of us cherish,
Mm,
and with the generations becoming different, there is right now um, a lot of hesitation in really getting too much exposure to electronics and too
yeah.
much exposure to very shallow and quick ingestion of content, right? Because in the past, when let's say me and you were training, right, the you really had to read the whole paper. You couldn't read the highlights,
Yeah.
and also you couldn't rely on AI to tell you what this article
Yeah.
is about.
Yeah.
You really need to read it all and really understand it. And that's part of developing sometimes the skills in critical thinking, right? That you need to make your brain synthesize the information for you. Don't rely on synthetic information being delivered to you.
And ironically, given the fact that the increasingly more and more things that will be seen as routine as we go forward will be automated and will be AI
Yes.
driven and things like that, is actually that skill, that discipline that will be required, right? It's the
Yeah.
how to think, not what to think,
Yeah,
that's going to
exactly.
be important.
And
Um,
this might be where, the future science careers will go,
mm,
right? Because how to be creative and create some unique content or unique way of thought pattern, that's where, scientists will be needed. They won't be needed
yes,
for routine data analysis. They won't be needed maybe even to formulate experiments based on input, but they will have to be there to probably guide the systems they will be interacting with to formulate the future of our research.
yeah, yeah, and you talked about early in your career sort of the learning about organizing different people from different disciplines and
Yes.
the management part of it, and I think there's almost an opportunity for people to develop some of those skills earlier on, right? Because you're gonna manage all these different GPTs to interact with each other, and actually, you don't have to be the expert, but you have to coordinate all this expertise. You have to coordinate all this
Correct. Yeah.
computational power, and point it at the right questions.
Yeah, exactly. And that's the thing which you can only get experience with by really immersing yourself with different opportunities, I think.
Mm,
So in your scientific career, right, I think the piece which is probably driven me to become who I am right now is this diversity of experiences,
right.
being able to engage in different areas of research, like change fields, go from, drug discovery to therapeutics to
Mm,
translational medicine back to drug discovery. That gives me certain level of, um, abstraction, right? So I can think
right.
about problems more broadly. And that's the unique opportunity that, many of us can create for ourselves. Those are the choices that we can be making, right? And
Mm,
for people who are very early, sometimes people approach the career development in a very fixed way, right? So they will
right.
seek, let's say, a coach, and they say, look, you need to have a plan, and the plan is like you decide on your field, and then you need to score certain goalposts, move
Yes.
from point to point, otherwise you won't succeed. In fact, most interesting jobs and opportunities I got were just by serendipity.
Yes.
They just happened. They fall on my lap, and I thought, oh, this is great. Let's do that. Right? There was never, maybe anything deliberate or anything, let's say, so well planned. And that's maybe something that also for many of us cause a lot of stress. Right?
Yeah.
Where am I in career? I'm making the right choice. As frequently people will start the careers for which they are not always a good match, and
That's true.
they're so deep into, let's say, the learning, so deep into getting this initial experience, they don't feel like making a switch.
Right.
And with the accessibility to knowledge, accessibility of training, I think making those switches should be way easier nowadays.
Mm, mm, mm. Yeah, it should be for sure. And it's interesting. Is that famous Steve Jobs speech, isn't there, about you can only join the dots in hindsight. You can't usually join them on the way forward from an experience and career
Yeah.
point of view. Uh, so we've sort of gone into this part of the conversation anyway, but in terms of sort of how your thoughts on how people should think about their careers, or your
Mhm.
advice to people who are perhaps early on in
Yes.
their careers. So you talked about I guess we're talking about being open to opportunity, right? Or being
Yes.
sort of accepting of different routes and not being too hung up
Mhm.
on this is where I'm going.
Yeah, no, that's true. That's, I think, the hallmark of what I think would be.
Mm,
Another thing which it's all maybe obvious is nowadays, to make really progress in science, you need to be very intimate with technology. You
yes.
need to track the progress of technology very closely, as new methods, new techniques, new hardware, new ways of doing stuff are developed essentially daily. right?
Yes.
And another piece which also comes with that is that due to the sheer scale of observations you're making and all the tools that you need to look and understand how they work, you can't really have, in my mind, successful career without acquiring some software and data science skills. That's where you need to be ready to take advantage of the tools that are being
Hmm.
developed, because everything becomes faster, more efficient, more precise, higher resolution, right? And that creates obviously opportunities for discovery that were not available before.
Yes. Yeah, and you don't want to wait for the proof that you get from your competition proving
Mhm.
it.
Yeah, no, you need to be fast about
Yeah,
discovering something nowadays.
yeah, absolutely. And this probably builds on what you've just talked about, but if you if you bumped into yourself, perhaps, getting off the plane in Chicago for the first time,
Yeah.
or heading out to Grail for the first time, or whatever it
Yes.
might be, whichever point makes most sense, and you had the opportunity to give yourself one piece of advice, what do you think that
Oh,
would be?
um, I would say, I would tell myself that no job is too small, no
Hmm.
problem is too small. Continue with gaining your experience and continue going, right? Don't get fixated maybe on your ambition, on how fast you're progressing what you do, right? And that's the thing, which we talk about,
Yeah,
connecting the dots, or thinking about yourself in retrospect, right? Because many things that had good things that happen really happen, as I mentioned to you, by serendipity,
yeah,
right? And there's nothing that I can point a finger at and say, yeah, I forced it to become it, right?
yes,
And that was it. Relax a bit and enjoy the ride.
yeah, yeah. I think this is a common challenge for ambitious people.
True.
With two things I think that come from it is that, sometimes you get so caught up in where you're trying to go that you forget about, oh, well, I actually have to do a really good job with what I'm doing here to get to that next stage. And I know I've had that backwards in my career at times. And I guess the other part of it is that someone told me once that, career doesn't just go up
Oh,
in a straight linear progression. It goes in plateaus. You go very quickly
right.
sometimes, and then you'll have a long period of time where you don't progress very much because actually your skills and your capabilities and your experience are catching up to where
Hmm.
you've got to. And until they do, and until you get to the point where you're actually,
Right.
in terms of your own development, beyond where you are currently, then you won't move on. But then when you do move on, it could be a really quick burst again, and then
Right.
you hit another plateau and this kind of thing. And I think those are kind of concepts that took me quite a while to learn. Not
Yeah.
theoretically maybe, but in terms of practical terms. But I think, this is a common tension for ambitious people, right? You want to go quick, but you've got to make sure you're
Yeah, of
present and
course.
engaged.
Well, and then you actually ready to move on, right? That
Yes,
you have
exactly. Yeah.
taken your time really to understand problems you're trying to solve, and then you have a lasting knowledge. And I think the most, this is also like truism, but the most lasting knowledge is from failure,
Right.
right? If you were ambitious and always successful and never failed, what would you would have learned, right? So that would be
Yes.
also another advice to myself, if you ask me.
Yeah. Okay. Okay. Embrace the failure a little.
Exactly. Yeah,
Yeah.
because the lot of good discoveries in science are actually made is like, you look at, oh, it failed. But that's
Mm,
interesting.
mm. Yeah, exactly. And I suppose, in an industry where you are going to encounter a 95% failure rate, you've got to get pretty comfortable with that pretty quickly, right?
Yes, it's true, right? And then the grand challenge is like how to make the drug discovery programs, for example, fail less.
Yes.
And
Yeah.
that's where I feel like the new advances in the field are really heading to how to make more thorough decisions driven by data. This is where ML obviously can come to help. And then how to increase quality of our observations, how to build models of disease that translate better, how to go through design of your trials, how to go through even approval process, how to build a molecule with simplicity in mind
Yes.
that is more likely to win in clinic.
Yeah.
And those are the challenges that many companies are right now addressing. And a lot of the development that I see, even in Cambridge, US, is related to building platform companies that tackle those new challenges, or
Yeah, I
maybe
think that's
old
a
challenges.
yeah, yeah, exactly. Embedded challenges, I think. And I agree. I think, um, I've seen more and more of these conversations happening over the last few years as well. And I think technology has enabled people to think beyond this is the program we're developing, and among um, the old sort of frame of reference, the old paradigm, right?
Mhm.
The old way of doing things, and look at, well, actually there are systemic issues here that aren't anyone's fault. They are a feature of the environment we've been working in. But with the technology we have, do we have to continue working in these ways? And the answer is probably no. There's lots of there's lots of hurdles to overcome on the path to that, of course. But it's good that people are starting to ask those questions. I think that's a good place to start.
Oh, excellent.
Yeah. And Martin, look, going back to Metaphor for a
Yeah,
second. So we
of
talked
course.
about the work you guys are doing. We talked a little bit about where you're up to with it. Tell us a bit as you can. I appreciate some things will be confidential about what's next.
Yeah, so what's next? Well, shortly after launching about a year in, right, we managed to attract a partner. We started our collaboration with Novo Nordisk that the deal
Hmm.
is valued at roughly 600 million. I consider this an adventure and also a tremendous opportunity for Metaphor,
Yes.
because now we're gaining visibility, we're getting a lot of interest, and this pushes us to further build up our therapeutic discovery pipeline, further advance, the platform, obviously with support of Novo Nordisk, but also
Hmm.
independently. And then how to garner more interest from pharma companies. We're getting, obviously, I said, a lot of interest. I'd love to see us developing our partnership model further.
Right.
And with the build out of the pipeline, what I mentioned to you, like working with cardiometabolic space and also inflammation immunology, I think there
Hmm.
will be a lot of opportunities to do this. So the balancing and of investment between platform development and pipeline is going to be key because the technology obviously is budding. There
Yes.
are a lot of advancements in ML field and AI tools that we need to really be taking advantage right now, and we're doing that. And then on the other hand, to really prove the value to investors, we need to be creating a curative medicines for patients,
Hmm.
and deliver on the promise of the mimicry I was describing to you.
Yes, absolutely. Well, plenty to be getting on with, and, and we wish you the best of luck with it, Martin. Thank you so
Oh,
much
thank
for
you.
joining us.
Yeah, it was excellent. And thank you. And looking forward to maybe talking to you some later.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.